Hello dearest viewer, today we will be exploring if it is possible to grow an entire ecosystem out of an algae cookie. Now, of course, this requires a bit of background. For about 3 years or so, I've had this vase sitting on my windowsill filled with just a bit of pond water, not much else, and in that pond water algae started to grow, of course, and there were a bunch of critters, and I never really filmed it because it was just a personal project, something interesting. And at some point about a year ago, for whatever reason, maybe I was on a holiday or something, I stopped watering it because it evaporates like crazy, of course, especially in the sun. And then it had a water level up till here or something, and I decided, you know, let's see what happens if it really dries out. So I've been drying it out for a year, and remember, and the only thing that was in here was pond water, all the critters that came with it, and all the algae that started growing out of it. And I think I added one Java fern at some point for my aquarium. And in the baking sun, it condensed into this little disc of dried algae and hopefully some eggs of some small crustaceans. It turned into an algae cookie. It's very dense. It's like actually a chunky thing. It smells a bit like a rocky shore on a beach, very algae-like. Look, a snail. It tastes kind of salty and algae-like, but not bad, just salty. And apparently there's also some sand in there. And I guess we'll just add some water and see if we can grow an ecosystem out of this.
So here it is, the algae cookie in all of its glory. Looks quite funky, right? If we take a bit of a closer look, we can see much more details in the cookie. There are several individual snail shells visible. The different colors and structures visible show us how there were several different plant and algae species present, which dried and potentially even oxidized at different rates. You can also see that there was some mold growing on some of the algae in the later stages of evaporation, just before it all became completely desiccated. The grayish-bluish colored stuff, which itself is now also completely dry. It looks very detailed and intricate so up close, almost like an alien landscape. Anyway, I think that you get the picture, the cookie picture. Time to actually add some water. So in goes the cookie, as well as the snail shell that fell off when I bit it. And the water. As the water penetrates and wets the cookie, air bubbles are forced out. If you look closely right under the cookie, you can see that the light is refracted differently through the water there. Just like when you mix cold water with hot water, or when you see hot air rising from asphalt or the roofs of cars on a warm day. In this case, it happens because some substances in the cookie are instantly dissolving in the water. Quite interesting. It's not quite as visible on camera as in real life, but after 1.5 hours the water got a slight tint to it, and the water near the cookie has become a lot darker, almost like tea. The cookie is still solid but not rock hard anymore. It has the consistency of stiff leather. Let's mix the water a bit. A day later, the denser water was dissolved. Stuff from the cookie sank to the bottom again, and on top was a bit of a cloudy layer, which looked really cool. The cookie still feels the same. Another day later, the entire water column got cloudy, and 2 days later even more so. 1 more day later, so 5 days since the rehydration, I noticed something interesting. Just above the waterline, on the glass, throughout the entire vase, were these little red mites. There were a lot of them. Did they emerge from eggs in the algae cookie? Perhaps, but then why would they all be above the water and not a single one in the water? That's strange, right? I also did not see a single mite on the outside of the vase or on the surfaces next to the vase. So they must be coming from the cookie, I guess, but aren't aquatic? Very strange. I don't really know what's going on. To make it even weirder, they are all dead. All of them. And quite a lot of them appear to be missing limbs. Looking back at the footage, it appears they emerged at the same time the water became hazy, so 3 days before this. This means that wherever they came from, through presumably the algae cookie, somehow, they all emerged and died at the same time in the span of 3 or 4 days. That is so strange. I guess one could sort of consider this an ecosystem, though I am so perplexed by this that I won't count it yet. So we'll continue the experiment, as you might have been able to guess by the progress bar of your video player. This is not the first time one of my projects presents me with a mystery, and I'm sure it won't be the last, but as far as mysteriousness goes, this one is definitely up there. I find it so cool how these tiny little worlds in glass, easily overlooked, can sometimes tell such strange and interesting stories. But it's way too early in the video to already start becoming philosophical, so let's quickly continue. Another 6 days later, the water had become much more cloudy, which at this point is probably the result of a very high concentration of bacteria in the water, which can thrive due to all the nutrients being released in the water by the algae cookie, and a complete lack of predators and even competitors. They are sort of the pioneer species of our re-emerging ecosystem. There's a slight chance that they came from outside the vase, as this experiment is not being conducted in a sterile environment, but they most likely came from spores or cysts within the algae cookie. More evidence for microbial life comes in the form of biofilms growing on the water surface, as well as the sides of the glass. Something else I've noticed, and this fortunately almost never happens with my experiments, it kinda stinks. Only if you really stick your nose in it though. 2 days later, all the microbial growth has increased, and unfortunately also the smell. So in a life in jars first, I decided to move it outside, so now it can stink up the city. Well, it took 15 days, but it finally doesn't smell anymore, so it can come back inside, exactly 4 weeks after the start. It might not look like much is going on, but the large amount of biofilm that accumulated on the bottom and is still lining the water surface and the sides of the vase tells us that there has been a lot of microbial activity. The horrible smell also tells us that, so I'll add some water to compensate for the evaporation, and then I think it's time to finally, take a bit of a closer look. Here at the lowest magnification, against the dark background, we can instantly see that there is lots of microbial life zooming around. These large microbes you see zooming around are flagellates, though I can't really see what kind. The hazy color in the water column might have been caused by these cells, but more likely that is the result of bacteria. If we go to a higher magnification, you can actually see those bacteria. It might be a bit difficult, but all the very small black specks you can see are individual bacterial cells. If we look closer at the flagellates, we can see that there are a lot of different shapes and sizes, so there are probably different species of flagellates present. Here you can also nicely see some of the bacterial cells moving around. They are surprisingly mobile for their size. Just imagine these were large animals. They would be so fast. Or don't imagine it, see if I care. Anyway, for the water to be this cloudy, there must be a lot of cells present. If you thought this was a lot already, well, think again. Here, right next to an air bubble in the microscope slide, you can see the true insanity of the densities that these bacteria reach in our freshly rehydrated ecosystem. I'm not entirely sure why they seem to be moving so much more right on the edge of the air pockets, but it is interesting. Maybe it has something to do with different concentrations of certain chemicals near the air, making them go crazy. These are the kinds of densities that can make the water cloudy. We're talking about an insane number of cells in even just a single drop of water. They are all supported by the nutrients that have and are still leaking out of the algae cookie. When you have an aquarium that you overfeed, this can also happen. If the water turns cloudy, that is most likely because of bacteria in the water feeding on the nutrients provided by all the fish food that isn't being eaten by the fish. So you, or more likely the people you asked to care for your fish while you were on a holiday, might have fed too much. But I digress. These bacteria are currently forming the base of the young emerging food web. They don't face much predation yet, but are likely being predated on by at least some of the other microbes. At some point, the ecosystem will inevitably switch to one where photosynthetic organisms, so algae or primary producers, will form the base of the food web. But now we're seeing the earlier pioneer version of the food web, a month after the algae cookie was rehydrated. That is so cool! So, turns out that there's already loads of microbes living in just the water column. This raises the question, what lives closer to the cookie, near the bottom of the vase? Well, let's find out. Somewhat surprisingly, it doesn't really look that different. It's pretty much more of the same. Maybe we should take a direct look at some of the biofilm and even at the algae cookie itself. We'll have a bit of a full slide, but that's fine. We are immediately presented with a clue to one of our mysteries. Here we have one of those mites we saw earlier. It was in the algae cookie, so that suggests that they did in fact emerge from it. It doesn't solve the mystery by any means, but at least we now know that there are, or at least were, also present in the water. This one is also dead though. Maybe they were in the vase before it dried out and their corpses started floating upward after the water was added again. That still leaves the question of how they emerged above the water surface though. At a higher magnification, the mite itself isn't much more interesting, but we can see something else. A few microbes of approximately the same size and shape. Not sure if they are dead or alive though. Not sure what they are, but they look to me like Euglenids that aren't feeling so good. Apart from the mite and the Euglenids, the microbial community looks very similar to what we saw before. So this is funny and silly. I decided to measure the pH at this point, thinking that would give us some interesting information. But I didn't write the value down, and now that I'm editing this video, I forgot it. And I also don't have access to the color chart that came with these strips, so this is completely useless right now. Of course, I could have just not included this clip, but I wanted to show you that, contrary to what most of you probably believe, even I am not perfect. Alright. In the next 2 weeks, the base of the food web indeed started to shift, and it seems that by now our renewed ecosystem is mostly dominated by algae and other primary producers. The algae cookie is covered by a film of photosynthesizing cyanobacteria, while there's green algae growing on the glass and in the water column, resulting in the green tint. Over the next week, this algification continued, with the cyanobacteria becoming more dominant. You can even see bubbles on the surface of the cyanobiofilm, which are oxygen bubbles. Let's take another microscopic look at water and a bit of algae near the bottom of the vase. Here we have a nice dark field view of some of the algae. Quite pretty if you ask me, and very green, meaning there's a lot of healthy algae. Looking even closer, we can make out some of the individual cells. Almost all the algae we see here are green algae, and there's also some other microbes zooming around, some kind of fast-moving ciliates, I think. So which of the green algae do we, or I guess, recognize? Because this patch of algae is so dense and the cells and/or colonies overlap so much, it's somewhat tricky to recognize them. And because for some reason I was moving around so much while filming, it's a little difficult to point out to you what we're seeing, but I'll try to manage with arrows and circles. So bear with me. This is a Cosmarium, a type of green algae. Then there's a bunch of what looks like small colonies of algae. Unfortunately, because I can't really see where one group ends and the other starts, and because I'm an amateur, this means I cannot really figure it out. But it could be maybe Pandorina or Eudorina. Probably not though. What it almost certainly isn't is Merismopedia. But the reason I mention it is because I just quickly wanted to introduce you to this genus of cyanobacteria, if you didn't know it already, because it's super cool. The cells divide in 2 directions, which results in these very structured and rectangular colonies. That's pretty neat, right? Just look at them. So what else can we recognize? These cute little colonies are also green algae in the genus of Scenedesmus, which always create these little colonies of, depending on the species, usually 4, 8 or 16 cells. These are also Scenedesmus. Here we have something that isn't a green algae. It's a diatom, with a glass house to protect itself. This looks like it might be a Chroococcus, a cyanobacteria, or blue-green algae. I am tempted to say that some of these grids just might be Merismopedia, but I'm really not sure. I hope you could somewhat follow this. 3.5 weeks later, 2.5 months after the cookie was rehydrated, it looked like this. Visually, not much more has changed, apart from some water evaporating and all the algae becoming a bit denser. The biofilm on the water surface also became denser, and I tried to make a hole in it because I didn't necessarily want to sample that, but it didn't really work out. In order to once again sample the water near the bottom, which if you're wondering why I'm doing, because I'm hoping to have a denser but still representable sample of the entire vase, I had to push the algae cookie away, which was still in one piece, though quite a bit softer. And I hope you're ready for another round of algae identification, because here we go. Here we see a lot more of the Scenedesmus colonies, as well as some individual diatoms. There's also a lot of bacteria zooming around in the quote-unquote background, and we can see some flagellates as well as some really fast ciliates. There's also more of the dense, unidentified algae film. This is something interesting. The structure you're seeing here is a hyphomycete, a kind of fungi, which we don't see too often here on this channel and is an entirely different domain of life. So that's quite exciting. Here we can see a bit of the biofilm that was on the water surface, which turned out to be quite interesting after all. All the tiny black shapes very close to each other are individual bacteria, which together are forming this thin biofilm. Pretty neat. I think it's strangely satisfying to look at all these bacteria zooming around seemingly completely random throughout the water. It's pretty fun to look at. But maybe that's just because staring at algae through a microscope for such a long time caused weird changes in the structure of my brain. Who knows? Let's add some water again to compensate for evaporation. And as it turns out, this is accidentally the last I filmed of this ecosystem.
Well, that may have been a little bit of an abrupt ending, but I actually temporarily moved to Sweden for some research, and as you can imagine, I couldn't really bring this project with me. I've tried to recreate the setting of the intro, but with limited success. However, to answer the question if it's possible to turn an algae cookie into an ecosphere, I would say yes, it is. Now, it might not be a very flashy ecosystem with loads of different organisms and a lot of trophic levels and apex predators and stuff like that, but we still saw a lot of diversity in algae and even larger protists like ciliates and flagellates, and also many bacteria. So yes, an ecosystem indeed. And over time, we might even see the emergence of larger organisms or even animals as the rest of the algae cookie, especially the center, gets even wetter. But we might also not. Of course, I will post an update about this project as soon as I'm back in the Netherlands. I think this was a very interesting experiment, and I hope you liked it too. I would like to thank Daniel Webb and Andrew Glass, as well as all the other patrons for their generous support. Between my studies and this YouTube channel, I don't have much time to work, so your contributions provide a very welcome, steady stream of some extra income. I really appreciate it, so thank you. And well, that is the story of the rehydrated algae cookie turning into an ecosystem, so far. It showed us some beautiful examples of the tiny forms of life that can be found in aquatic ecosystems and which are evidently very resilient. Thank you very much for watching, and goodbye.